Sorry, it got mistakenly stopped. So I was telling you these coat protein molecules plays important role in the formation of vesicle. So here COP2 involves in the formation of vesicle at the endoplasmic reticulum, and these particular vesicle are meant to go and fuse with the Golgi cisternase. So wherein for the form modifications of the protein molecules, and COP1 is going to be involved in the transport of proteins in a retrograde direction, that is from the Golgi complex to the endoplasmic, that is rough endoplasmic reticulum, because there are few proteins which are, as you have already we have studied, which are going to be called as endoplasmic resident protein molecules. Means they are need to function in the endoplasmic reticular membrane within the lumen of endoplasmic reticulum. So these protein molecules are going to get mobilized through COP1 vesicles. Then we have the clathrin. So clathrin vesicles transport the proteins from the plasma membrane and the trans Golgi network to late endosomes. Means clathrins are going to be very important set of coatomers, because these protein molecules involves in the formation of vesicles at the secretory pathway, endocytic pathway, and as well as intracellular transport of vesicles, wherein the formation of vesicles are mediated by the clathrins. So the trans-Golgi to plasma membrane vesicles get formed by these clathrin. So you can see electron micrograph wherein the vesicle formation is being shown. So if you come to how exactly vesicle formation takes place as an overview, you can see here. So the green color protein molecules, which are nothing but the coatomers, they are going to polymerize at the membrane. While polymerizing at the membrane, they are going to interact with the cytosolic part of the receptors, and these receptors are the one which will be interacting with the cargo or the protein at the other end. So now these particular receptors, are the cytosolic end of these receptors, will get interacted with the coatomers either directly or with the help of an adapter protein molecule, and thereby when these coatomers they start polymerizing, they are going to drag these receptors. While dragging these receptors, they also drag the portion of the membrane inside these particular polymerizing coatomers, and thereby they starts creating a small neck of this particular part of the membrane, finally which get due to the polymerization of these proteins get pinched off, and the whole of this particular portion becomes a coated vesicle. And here proteins like snares are also going to play an important role in this process. And you can see on the other hand, so there are fusion, so wherein the proteins are these particular vesicles which are carrying the cargo are nothing but the proteins or the other molecules. They are going to get transported to the place of fusion or to the target membrane. This mobilization will happen with the cytoskeletal elements, and once the vesicle reaches near the target membrane, so these particular vesicles on their surface, they are going to have lot of proteins called snares. So these snares are going to be fibrillar proteins, and when the two membranes almost they coming near to each other, and what happens is that these snares are going to form a ternary complex. Means they start winding around each other and bringing both the target membrane and the vesicle membrane very near to each other, and finally due to the hydrophobic collapse, or other way we can say due to the hydrophobic interaction of the the lipids of the plasma membrane and the target membrane and the vesicle membrane. So they are going to get fused, and the cargo or the content which is present within this particular vesicle will get into the other side, and whereas the portion of the membrane of vesicle will get fused with the target membrane, and which can get recycled through the formation of vesicle. So now this particular vesicle formation, as I told you, uh, it's not a very simple or an easy step. So for the formation of vesicle, it requires a lot of other protein molecules help, and these cautomers themselves, they are not going to be monomeric units. They are going to be having a number of subunits sometimes, or there may be composed of several other protein molecules. So COP2, for example, it is made up of Sec23, 24, uh, one dimer, and there is Sec13, 31, one dimer. Together with Sec16, they form the COP2 complex. And here, for all these process of formation of vesicle, or in other words the coating of vesicle, it requires the energy which is provided by the GTPase activity of a set of proteins called GTPase proteins. And in case of COP2, the GTPase activity is done by SAR1, and in case of COP1 and and clathrin, so COP1, it is going to have the cautomers containing 7 different subunits, and ARF is going to act as an GTPase protein molecule, whereas in case of clathrin, it is a combination of protein called clathrin and the adapter proteins. So it can have clathrin plus adapter 1 protein, AP1 complex, where ARF is going to be GTPase. See here, different type of adapters are going to be utilized by clathrin for at different vesicle formation and at different target membrane where vesicle formation takes place. For example, clathrin binds with the GGA protein molecules in trans Golgi, where the vesicles get formed for the formation of endosomes, like AP2 at the plasma membrane, AP3 at the Golgi to lysosome, melanosome, platelet vesicles, like that. Okay, sorry. So here these particular vesicles which are going to be involved are formed. So the formation of vesicle is going to happen, as I told you, by the process of polymerization of cautomers, and this polymerization of cautomers is going to get initiated by a particular set of protein molecules, so which are going to be involved in the polymerization. Sorry. So here you can see this process of polymerization of protein is going to get initiated by a protein called as ARF. So this ARF protein is a cytosolic protein molecule by itself when it is inactive, because this is a nucleotide binding protein which binds with the GTP, and when it is bound with the GTP, it is going to be an active protein molecule. When it is going to be bound with the GDP, it is going to be in inactive nature. So this type of protein, we find several within the cell which are going to be functioning during various kind of regulated activities. So now here ARF, which is initially an inactive protein molecule having GDP in its nucleotide binding pocket, so will get activated by the exchange of GDP with GTP, which in turn is achieved by the help of a protein called ARNO, and this ARNO is going to be an GEF, that is guanine nucleotide exchange factor or protein, and this causes the activation of ARF, and this ARF activation leads to binding of ARF to the membrane. This ARF is going to have an hydrophobic chain, a palmitic acid chain, which is actually attached, which is covalently linked with the this particular linked in the protein, and when it is going to be in cytosolic free flowing free protein molecule, that time this particular hydrophobic chain will be present within the core pocket of the protein. But with the exchange of nucleotide, this particular hydrophobic chain will get released, and now this particular hydrophobic chain will get hooked up or get inserted itself into the membrane of into the target membrane, or the membrane at which the vesicle formation is needed, and this whole process is called as priming. So once the priming is done, then the adapter proteins are get recruited. These adapter protein molecules in turn they start recruiting the clathrin, and followed by that there will be uh, binding of cyanamine, which is going dynamines are going to cause polymerization and helps in the pinching. Then further removal of the cautomers takes place by the chaperonins. So I'll do one thing, the whole process I'll explain in the next video. So thank you, hope you people are following, and please do ask any doubts in case if you get when you're going through the topic.